Okay, so let's talk about energy for life. This is chapter 7. So what we're going to focus on today is previously we talked about energy for life, which is about how plants go about doing photosynthesis in order to make energy source not only for self, but obviously for us consumers, right? The ones that actually consume the plants. So today, let's talk about energy for cells. So once we consume, let's say a carrot, how do we go about using that energy so that we can actually fuel all of our cells? So the topic we're going to talk about today is cellular respiration, glycolysis, which is what happens outside of the mitochondria, and then within the mitochondria itself, how do we go about breaking these different molecules down in order to actually get energy from it. So let's start off with cellular respiration. So when you hear this term, like, what are you thinking of? We see the word cell, so something to do with cell, and respiration. What do we tend to think of when we hear the term respiration? Well, I tend to think of breathing, right? So in a way, you could think of like, how does the cells essentially breathe in a way? It's not quite an exact interpretation of it, but why do we breathe? Like as humans and other organisms, why do we breathe? And it's because in order for us to actually take that carrot that we're eating, oh look, this is by coincidence. I forgot she was also eating a carrot. But anyway, by us consuming this carrot, how do we actually take that carrot and make it into energy, right? So we actually have to breathe in oxygen so we can actually convert the glucose that's actually found in this carrot and convert it into a usable energy source that our cell knows how to use. Okay, so what is the purpose of cellular respiration? To produce ATP, which, if you recall, that's our energy currency, right? So that's what the cells, that's the structure that the cells can actually use for energy. And in order to do cellular respiration, we require oxygen as well as some sort of glucose, so some sort of energy source. And the output of this is carbon dioxide and water. And so this is why we breathe. So we breathe because we require that oxygen, and we'll see why later on in the talk. But we require oxygen, and then as a byproduct, we actually produce carbon dioxide that we're going to exhale out. And so if you think about photosynthesis, this is kind of like the reverse of the photosynthesis. So in photosynthesis, we took carbon dioxide, water, and sun, right, to make energy, and then the byproduct of that was the glucose and the oxygen. Well, in this process, we're reversing it. So we're actually taking oxygen and glucose, and then the byproduct from that reaction is the carbon dioxide and the water. And we also make energy. Okay, so think about respiration and just things for you to think about as we're going through this talk, right? What is it? So the idea of like breathing in and out. So what are we breathing in when we do breathe in? You know, what are we breathing out? And then your cells, are they doing the same thing? Well, you know, they're doing something really similar, right? So our cells are also breathing in oxygen, using up that oxygen for this process, and then expelling or exhaling carbon dioxide. Then that oxygen, that carbon dioxide will eventually travel through our system, and then we breathe it out, right? So cellular respiration is the reason why we breathe. Okay, so two type of reaction that you can think about as we're talking about this is oxidation and reduction. So what I want you guys to recognize and appreciate from the oxidation and reduction during cellular respiration is that in oxidation, what's happening is we're removing hydrogen atoms. So as we're removing hydrogen atoms, we're losing electrons. So you can see here in this reaction on here, we have glucose, right, which has CH and O, and as we oxidize it down to carbon dioxide, you can see that we lost hydrogen atoms. And in the process of losing hydrogen atom, we also lost the electrons that go with the hydrogen atom. Whereas in reduction, we have the opposite. So in reduction, we start off with oxygen right here, right? We reduce, and when we reduce, this pen is not working super well. We actually added hydrogen atoms, and when we added the hydrogen atom, we also gain electrons. All right, so whenever you see oxidation, just think loss of hydrogen atoms, loss of electrons. Whenever you see reduction, just think gain of electron, gain of hydrogen atoms. Okay. So that's kind of if you want to learn more about this, you'll learn more about it when you take chemistry. But we're just gonna think of it that way. So as I'm talking about different reactions, and I'm talking about whether a substrate is being reduced or oxidized, think of the loss and gain of electron that is accommodated with that. Okay, so let's briefly kind of look at an overview of the breakdown of glucose in cellular respiration, and then we'll start actually breaking it down into pieces. So the breakdown of glucose has 4 phases, right? We have glycolysis over here, and notice that glycolysis, so this is the mitochondria, this big structure right here is the mitochondria. Notice how glycolysis is outside of the mitochondria, so I'm going to write outside, right? And so glycolysis is when we first take glucose and we convert it to pyruvate. And we're not going to talk about it too much, but what I want you to know is once we have pyruvate, this is just the start of the cellular respiration. But this pyruvate will go through this whole process right here with the assumption that we actually have oxygen at the end. Okay, so I'll just put oxygen here. So it's with the assumption that we actually have oxygen at the end. If we do not have oxygen, we're not going to talk about in depth, but we will point it out in a little bit. If we don't have oxygen, this pyruvate will go through 2 other processes that are anaerobic. So anaerobic just means that it does not have oxygen. So what I want you guys to appreciate is at this point, when we finish glycolysis and we get to pyruvate, that pyruvate can either go down the aerobic process or anaerobic, and this all depends on if we actually have oxygen available, right? After that, if we do have oxygen available, it's going to go to this preparatory state, which is converting this pyruvate and making it better accessible. So taking the pyruvate and converting it to a different type of molecule that will make it more accessible, and from there going through a citric acid cycle. So here's where we're going to try and collect some electrons from this molecule that's here. And then the last part is taking it to the electron transport chain. So in the electron transport chain is when we're actually going to really use the electrons to drive what we'll talk about later is known as the proton gradient, and driving that in order to make energy. So the end goal of this whole glycolysis is to make this huge amount of energy, this ATP. You can see there's 34 molecules. We're making ATP throughout. We're making it here and we're making some here. But 2 and 2, that's nothing compared to the amount that we make here in the electron transport chain. So let's kind of break down these 4 phases in a little bit more detail. So the first one is glycolysis. So again, this happens outside of the mitochondria. And in glycolysis, which occurs in the cytoplasm, so outside of mitochondria, what happens is we take the food that we consume and harvest the glucose, and we break down that glucose into 2 molecules called pyruvate. So glucose, if you recall, is a 6-carbon sugar, right? So we're basically going to take that 6-carbon sugar and break it into 2 3-carbon sugar. So this requires energy for us to actually break this down, but we're also going to make some energy. So the end result is we actually are able to make some energy that our cells can use later on. But in order to actually do this whole breakdown of glucose to pyruvate, it actually also takes energy. And this occurs in both prokaryotic and eukaryotic cells. So the investment part is when we have to actually use ATP. So remember, ATP is our energy source here. So we're just going to write energy right here. So ATP is our energy source, and we're actually going to use some ATP to break this 6-carbon sugar into 3 carbon. So here we're actually taking the phosphate from ATP here and harvesting energy. So we're going to get a lot of energy here. And we're actually going to add these phosphate onto this glucose molecule. So you can see one right here. We also have one right there, right? So we're going to transfer the phosphate. The phosphate, when it gets transferred, and this happens to a lot of molecules in the cell, when we add phosphate, we kind of activate it. So you can think of like the phosphate activating these molecules. So we took the 6 carbon sugar, added the phosphate, activated, and now that it's activated, it's ready for the next step of glycolysis, which is actually to convert it into pyruvate N. So first we start off with just a 3 carbon, and we're going to go through steps, all these different steps. You don't need to know all the different steps in full detail. What I do want you to appreciate from this is that you have these energy steps here. We're actually making energy. So here we use energy. So we're going to say that we use energy here. I know this pen is really thick. I don't know how to make it thinner. And here we're actually gaining energy. So we're going to gain some energy. So glycolysis is also known as the substrate level ATP synthesis. Why? Because our substrate is our glucose right here, and we're actually synthesizing ATP, but we're not using the whole electron transport chain yet. So at this point, where is the converting of the substrate of glucose into the pyruvate that results in this production of ATP. And so we have a net gain of 2 ATP. So where did we get this from? Well, we have 2 ATP here and 2 ATP here. So that's 4 ATP, right? Minus 2 ATP from up here. So minus 2. So 4 minus 2 is going to equal to 2. So this is how we end up with a net gain of 2 ATP. Additionally, we're going to convert some of that electron and protons from the substrate, and we're going to have it actually captured by NAD+. So I do want you to recognize that we are making an NADH here. We'll talk a little bit more later, but for now, just recognize that this NADH now has electrons and protons that we can then later use later on in our whole electron transport chain. But to kind of recap here, what I hope that you see is that with glycolysis, that the net yield again is our goal is to get those 2 pyruvate, which is going to be used next in a preparatory state, that we had used electron and hydrogen in order to make NAD plus into NADH. And this happens twice, right? Once for each one of those 3 carbons that we're going to convert into pyruvate. Additionally, we're going to make some substrate level ATP. So we're actually going to, while we do use some ATP, our end goal is to actually make ATP. So while we did use 2 ATP, we also made 4 ATP, which gives us the net result of 2 ATP. And so the next step, like I mentioned before, is really dependent on if oxygen is available. If oxygen is available, pyruvate is going to enter the mitochondria, and we're going to continue down to cellular respiration. But if oxygen is not available, then we're not able to have pyruvate actually enter the mitochondria to do cellular respiration. Instead, it's going to undergo reduction. So remember, we talked about oxidation reduction reaction. We're actually going to reduce pyruvate, and we're going to undergo 2 other type of processes because of the inavailability of oxygen. So I'll link this video up with the YouTube so you can watch it. But before we dive further into this whole process of cellular respiration, I do want to mention that, like I talked about before, after we do glycolysis, at that point, this pyruvate molecule is deciding whether or not it wants to go into the mitochondria, and it will if there's oxygen available. But if there's no oxygen, it undergoes what's known as fermentation. And so there's two fermentation that you guys are probably familiar with, and we're just gonna briefly touch upon both of them, right? So the first one is the one that happens, you know, those scenarios where school made you like run a mile, or they made you try to beat your best time. You're running, you're really out of breath, and you kind of get like a little cramp. If you're not like super athletic, you might have gotten a little cramp on your side. This is due to lactic acid buildup. So what's happening here is because you're exhausting yourself and you're kind of getting out of breath as you're running, right? You're getting kind of low out of breath. Your body still needs to make some ATP. So it can't undergo cellular respiration because there's not enough oxygen molecule for it to undergo it. And so what it'll undergo is it'll undergo this fermentation where it'll take the pyruvate, and because there's no oxygen, it still wants to make some ATP. So we're still going to make a net gain of 2 ATP here, but it's going to undergo fermentation and form these 2 lactate. Similarly, in bacteria and fungi, without oxygen, bacteria and fungi are also able to make energy, but they also undergo fermentation. And so that's what results, because of the fermentation, you result in making these alcohol products. And so similar to when we do undergo cellular respiration, in both cases we start off with glucose as our substrate, but in fermentation, let's kind of just summarize it, right? So this is going to happen in both us when we're forming lactate, but it's also going to happen in bacteria and fungi, where we're going to basically take this glucose molecule and convert it to either 2 lactate or 2 alcohol and carbon dioxide. This is the reason why beer has like that fermentation, or not fermentation, has the bubbles, right? It's the carbon dioxide. And we're also going to take NADH. I know I didn't go into that in too much detail. We're actually going to break up the NADH so we can actually consume the hydrogen and electrons, and so we actually can convert that into NAD plus. And previously it does take some energy, so we do use some energy. We use 2 ATP, but our net result of fermentation is that we actually will make some ATP, so we still end up with a net gain of 2 ATP. Okay, so this is the next step right here is going to go into the preparation state. So we'll stop here, and I'll see you guys in the next video.